Good afternoon, children. In our last video, we have learned about lac operon of chapter Molecular Basis of Inheritance. Today, we will start with the new topic, that is Human Genome Project. The genetic makeup of an organism lies in its DNA sequence, and if any two individuals differ from each other, then there should also be difference in their DNA, at least at some places. These assumptions led to the idea of determining the complete DNA sequence of human genome and consequently of Human Genome Project. It was a mega project. Human Genome Project was the mega project which took 13 years. For its completion, that is from 1990 to 2003. It was coordinated by the US Department of Energy and the National Institute of Health. The two main factors that contributed in the completion of this project are: number one, availability of simple and fast techniques for the determination of DNA sequences. Number two, genetic engineering techniques which help to isolate and clone any segment of DNA. Human Genome Project was called as a mega project because of some reasons, like the human genome is said to have approximately 3 into 10 raised to 9 base pairs. And if the cost of sequencing is $3 per base pair, then total cost would be about $9 billion approximately. Second reason, if the sequence were to be stored in a typed form, each page of the book containing 1,000 letters and each book containing 1,000 pages, approximately 3,300 books would to be needed to store the complete information of DNA sequence from a single human cell. The third reason is the large and enormous amount of data that was generated also necessitated the use of high-speed computational devices for the data storage, retrieval and analysis. Human Genome Project was in close association with the new and rapidly developing area of biology known as bioinformatics. That is a scientific discipline that encompasses the application of computing science and technology to analyze and manage the biological data. Now, goals of Human Genome Project. Number one, to identify all the genes, approximately 20,000 to 25,000 in human DNA. Second, to determine the sequences of the 3 billion chemical base pairs that constitute human DNA. Number 3, to store this information in databases in digital format. Number 4, to improve the tools required for data analysis. Number 5, to transfer the related technologies to other sectors like industries. And number 6, to address the ethical legal, and social issues that could arise from the project. There are some benefits from Human Genome Project. The knowledge about effects of the DNA variations among individuals can lead to discovery of new ways in diagnosis, treatment, and prevention of various diseases affecting mankind. It can help in learning about DNA sequences of non-human organisms that can lead to an understanding of their natural capabilities. For example, bacteria, yeast, Drosophila, which is fruit fly, plant, rice, Arabidopsis, then Cynorhabditis elegans, which is a free-living non-pathogenic nematode, have been sequenced. This can be applied to solve challenges in healthcare, agriculture, energy production, environmental remediation, etc. Now children, let us move to the next topic, that is methodologies of Human Genome Project. Two major approaches involved in Human Genome Project are: number one, expressed sequence tags. This method focuses on identifying all the genes that are expressed as RNA. Sequence annotation. This method involves sequencing the whole set of genome that contain all coding and non-coding sequences and then assigning functions to the different regions in the sequence. Now let us study about sequencing of genome. First step is, first the total DNA from a cell is isolated and converted into random fragments of small sizes. Then these fragments are cloned in a suitable host such as bacteria, yeast, using the specialized vectors such as bacterial artificial chromosome, that is BAC, and YAC, that is yeast artificial chromosome. The cloning of DNA fragments lead to the amplification of each fragment, which helps in easy sequencing process. The DNA fragments were sequenced using automated DNA sequencer, which work on the principle developed by Frederick Sanger. These sequences were then arranged on the basis of some overlapping regions present in them. This required generation of overlapping fragments for sequencing. The specialized computer-based programs were developed for the alignment of the sequences. These sequences were subsequently annotated and signed to each chromosome. The sequence of chromosome 1 was completed only in May 2006. This was the last of the 24 human chromosomes to be sequenced. The next challenging task was to assign the genetic and physical maps on the genome. This was generated using the information on polymorphism of restriction endonuclease recognition sites and certain repetitive DNA sequences called as microsatellites. Now let us study about salient features of human genome. There are 3164.7 million nucleotide bases in the human genome. In an average gene, there are 3000 bases. The largest known human gene is dystrophin, which have 2.4 million bases. Total number of genes in human genome are estimated at 30,000. Almost all, that is 99.9% of the nucleotide bases are exactly same in every human individual. For over 50% of the discovered genes, the functions are unknown. Less than 2% of the genome codes for proteins. Repeated sequences make up very large portion of human genome. These are stretches of DNA sequences which are repeated many times, sometimes 100 to 1000 times. They have no direct coding functions but help in understanding chromosome structure, dynamics and evolution. Chromosome 1 has the maximum number of genes, that is 2968, and chromosome Y has the least number of genes, that is 231. There are about 1.4 million locations in human genome where single base DNA differences occur, that is single nucleotide polymorphisms. This information is helpful in finding chromosomal locations for disease-associated sequences and tracing human history. Now let us study about uses of HGP or application and future challenges. Uses are: it diagnose, treat and prevent a number of disease or disorder that affects human beings. It provides clues to the understanding of human biology. Its knowledge is helpful in research involving biological systems including human biology. With the whole genome sequences and newer technologies, we can be systematic in our approach to various medical questions on a broader scale. All the genes in a genome, example, all the transcripts in a particular tissue, organ, tumor can be studied. So children, that is all about Human Genome Project. Now we will start with DNA fingerprinting. Before moving to the process of DNA fingerprinting, we will know about what is DNA fingerprinting. DNA fingerprinting is a very quick way to analyze the differences in the DNA sequences of two individuals. It involves the identification of differences in repetitive DNA. Repetitive DNA is a specific region in DNA in which a small stretch of DNA is repeated many times. Through density gradient centrifugation, this repetitive DNA are separated from the bulk genomic DNA. Bulk genomic DNA forms a major peak during centrifugation and the other small peaks are known as satellite DNA. These sequences do not code for any proteins normally, but they constitute a large portion of human genome. I am talking of bulk DNA. Now, the satellite DNA is classified into many categories such as microsatellites, mini-satellites, etc. on the basis of length of segment, number of repetitive units, the base composition, that is AT-rich or GC-rich. The satellite DNA sequences also show high degree of polymorphism and forms the basis of DNA fingerprinting. Now, what is polymorphism? Polymorphism is the variation in individuals at genetic level. Polymorphism arises due to mutations. It plays an important role in evolution and speciation. In a population, If an inheritable mutation is observed at high frequency, it is referred to as DNA polymorphism. There are different types of polymorphism, from single nucleotide change to large-scale changes. In an individual, DNA from every tissue, example blood, hair follicle, skin, bone, saliva, etc., shows the same degree of polymorphism. Thus, they become very essential identification tool in forensic application. As polymorphisms are inherited from parents to children, so it is useful in paternity testing. Now, the process of DNA fingerprinting. The process begins with a blood or cell sample from which the DNA is extracted. The DNA is cut into fragments using a restriction enzyme. The fragments are then separated into bands by electrophoresis through a agarose gel. The DNA band pattern is transferred to a nylon membrane. A radioactive DNA probe is introduced. The DNA probe binds to specific DNA sequences on the nylon membrane. Then, the excess probe material is washed away, leaving the unique DNA band pattern. The radioactive DNA pattern is transferred to X-ray film by direct exposure. When developed, the resultant visible pattern is the DNA fingerprint. Now moving to the steps of DNA fingerprinting or technique of DNA fingerprinting. Alec Jeffreys initially developed DNA fingerprinting, also known as DNA typing or DNA profiling, to find out markers for the inherited diseases. He used a satellite DNA as probe that shows very high degree of polymorphism and called it variable number of tandem repeats. The technique involved southern blot hybridization using radio-labeled VNTR as a probe. Now the steps, or you can say the technique, have the following steps. Number 1 is the DNA isolation or extraction. DNA is extracted from the cell in a high-speed centrifuge. Second step is amplification. Many copies of the extracted DNA can be made by the use of polymerase chain reaction. Then third is digestion. Digestion of DNA by restriction endonucleases. Fourth step is separation. Separation of DNA fragments by gel electrophoresis. Fifth step is southern blotting. Transfer of separated DNA fragments to synthetic membranes like nylon or nitrocellulose. Sixth step, hybridization. Hybridization with the help of a radioactive VNTR probe, that is small fragments of DNA, which help to detect the presence of a gene in a long DNA sequence. So, hybridization with the help of radioactive VNTR probe, which gets attached to single-stranded VNTRs having complementary nucleotide sequences. And the seventh step is autoradiography. Autoradiographic exposure to X-ray films. Detection of hybridized DNA fragments by autoradiography. This shows many dark bands of different sizes. A small DNA sequence is arranged tandemly in many copy numbers, which varies from chromosome to chromosome in an individual. The number of repeats show very high degree of polymorphism, as the size of VNTR varies from 0.1 to 20 kb. Consequently, after the hybridization with VNTR probe, the autoradiogram gives many bands of different sizes. Thus, these bands will give a characteristic pattern for an individual DNA. The VNTRs of two persons may be of same length and sequence at certain sites but vary at other sites. Therefore, it differs from person to person in a population, except in the case of monozygotic, that is identical twins. The VNTR belong to a class of satellite DNA known as mini satellite and are essential for DNA of fingerprinting. The sensitivity of this technique has been increased by using polymerase chain reaction. Now, applications of DNA fingerprinting: to identify criminals, to determine the true biological mother or father in case of disputes, to verify an immigrant really a close relative of a resident, to identify racial groups to rewrite the biological evolution. Okay, so DNA fingerprinting is used as a tool in forensic investigations, to settle the paternity disputes, to study evolution by determining the genetic diversities among population. So children, that is all about this chapter. We have completed the chapter.